So my name is Jens Hartung, and this I present here. Thank you very much for the invitation here. And I present a joint work from Hans-Peter Pifo and myself about the effect of missing values in multi-environmental trials on variance component estimates. Before I do this, I want to explain what my research background is. I'm study agriculture and doing since my PhD statistics in applying statistics on biological data and mainly on plant breeding side, plant size, and my focus is plant breeding data. The bigger the data and the more complex the models, the more interesting is the analysis. So, and I want to explain what are cultivar and evaluation trials. Afterwards, I want to speak about missing data and the classification there. Probably some of you know how to distinguish between the classes, but I want to explain what's the special thing in cultivar evaluation trials. It's slightly different to clinical studies or in animal science. Then I want to motivate my research, the work I want to present here, show you a simulation approach, show some of the results and make a conclusion in the end. So cultivar evaluation trials are field trials in the field where you grow some crops, probably genotypes which are planned to register from the official sites, and they are given to an official testing station, and at this station they test whether the genotypes are useful and have a higher yield. And this is normally done in small plots, which you can see here. You can see many plots. It's some randomized complete block design or some alpha designs are used where the genotypes are tested in 2-3 replicates in the field. So normally these trials are performed by governmental institutions. In Germany, this is the Bundessortenamt. I have data from Argentina, I think. The aim is to compare new genotypes submitted there from breeders, and they are normally evaluated for several locations in a target region. And for 2 or 3 years in Germany, for wheat it's 3 years, for most other cultivars it's 2 years. And after this testing process, the best genotypes are registered as cultivars, and only these cultivars are allowed by farmers to produce wheat so that they can grow this wheat. So the aim of the breeder is that the genotype is registered in the end, and in between there's some selection and only the best genotypes are registered. So I want to present how it's working in Argentina. We have a possible pool of wheat genotypes. You can see it on the left, and 40 genotypes are tested in the first year. And out of these 40 genotypes, some part of genotypes is selected, given the yield and other traits you were observed in the first year. And out of this pool, in the second year, the remaining positions are refilled, so that you have 40 genotypes in the second year again. And these 40 genotypes are tested again in several locations, and they are selected so that you keep the best genotypes in the third year, and you refill in the third year. And this is continued every year, year by year. And to explain what the missing process in this genotype testing scheme is, I would concentrate on 2 years. In the end, I analyzed 2 to 8 years, so all possible number of testing years. So if we have just 2 years, before I explain it on my data, I want to define the 3 classes of missing data. So just imagine that these blue quarters are the 2 testing years. And then we can divide the data in 2 parts. Green one is the observed data. Obviously, we have all data in the first year. And in the second year, we have only a part of the observed data, so it's part of the genotypes, and the red part is missing. So we have the genotypes on the x-axis and the years on the y-axis, and you see that round about 50% of the data is missing in the second year. So from these, so what we have is in mind a complete data set, but the complete data set is not observed. It's just a part of the observed, the complete data set is observed, and the remaining data set is missing. And depending on where the missing data depends on, we have 3 different classes. So if the missing data does not depend on the observed data or on the missing data, so it does not depend on your observations at all, so maybe just a sample is broken, then we call this missing data pattern missing completely at random. If it depends on the observed data, maybe due to selection, then we say that the missing data pattern is missing at random. And if we drop the data, or we would observe something, and that's why we drop the data, so it depends on the missing data too, as well as on the observed data, we call this informative missing. So maybe just think about of a field trial where you make a rating on biomass in mid of growing season, and there were some genotypes with very low biomass, and you see this is a candidate which has no chance. Then you delete this plot because of seeing what you see on the field. Then the missingness in the end of the year does depend on what you have seen before, and it depends. The genotype is missing in the final data because you would expect that something's going wrong. So there are 3 classes of missing data: missing completely at random, missing at random, and missing not at random. Coming back to the cultivar evaluation trials, where we have 2 years, then we can slightly change the presentation. So we have in the first year the candidates, 40 candidates. They are selected in the second year. Some of them selected. Other parts are not selected. And then we have genotype tests in the first year, in the second year, which are missing in the first year. So in some sense, the red quarters are missing in our complete data. So this is the complete data, and the green part is observed, and the red part is missing. And the question is, what is the data missing, the class of missing data, the data missing pattern? For this one, we took the genotypes of the second year exactly from the same pool as the genotypes in the first year. So we can assume that it's just a random choice of sort of taking the genotypes out of the pool. So this is missing completely at random. While the genotypes in the second year tested in the first year are not tested in the second year because they are not as good as the candidates selected. So they were dropped out because of the observations of the first year. So this missing data pattern is missing at random. And the question I want to focus on is whether this missing data pattern, the missing at random data pattern, has any effect on estimating variance components. The question is there any solution on this question? And obviously there is. There's a publication from Hans-Peter Pifo and myself with the old name in 2006, so a long time ago. And there we stated that missing at random can be ignored when you're using maximum likelihood, so when you're using REML as estimation method. I think most of you, whenever you're doing an analysis, you use REML as estimation method. It's state of the art for mixed model analysis. So whenever you use REML, you can ignore missing at random data pattern. It's published in Crop Science 46. In 2018 and 2019, another paper was published. It was also about the effect of missing values on variance components in multi-environmental trials. And they found a selection based bias on of data missing at random. And therefore the question was, is our paper from 2006 completely wrong? Have we misunderstand something, or what happened there in these two papers? And this is the motivation for me to look additional time on these problems where I already looked on during my diploma thesis. So coming back to our cultivar evaluation trials, the 2-year series, where we asked the question, is this a problem to have the candidates selected in the second year? So some candidates of the first year are missing in the second year. Is this a problem? And what we do is we simulated exactly this scenario, simulating genotypes, selecting them. And in a second simulation, having the same number of genotypes, having the same proportion of genotypes in the second year, but what we dropped is the selection. So we replaced the selection by just a random dropout. We randomly dropped out genotypes in the first year, and some of them are randomly tested in the second year, and some of them are not tested in the second year. So this missing data pattern now is missing completely at random, because the missingness of the data in the second year does not depend on the observed data in the first year, because the selection is done randomly. So what we do is we simulated data with missing at random, and we simulated data with missing completely at random. Just have in mind that when data missing completely at random, we think that the analysis should be fine and there should be no bias, everything should work fine. What we do is we calculated the relative error. So the relative error is just the difference between our variance component estimates and the true simulated value relative to the true value of the corresponding variance component. And if you do this for each simulation run and run 1,000 simulation runs, you can average across the relative errors. And the average relative error, this is what we call the relative bias. And as it is a difference, it could be negative if the variance component is an average estimated too small, and it is positive if the variance component is too large, estimated too large. So this is the main evaluation criteria we want to look at. We take 10% dropout, so we just dropped 10% of the genotypes from the first year till 90% of the genotypes. So in the one case, we keep 36 genotypes. In the other case, we drop 36 genotypes. We look at the data, what happens after one step of selection, so for 2 years, and we looked for 3 years till 8 years. And we also vary the size of the genotype variance. This has something, maybe we just need it in the discussion later on, but keep in mind that we have done this, that we slightly increase the genotype variance to see what happened then. And here are the results. What I present to you is now the variance component estimates for the genotype variance, genetic variance, genotype variance itself, the genotype by location, and the genotype by year variance component. And what you see is a relative bias of the genetic variances under missing at random. It's a scenario with selection, and missing completely at random, which is a scenario without selection. And what you can see from the 95% confidence limits, that the first value of the confidence limits is always negative, while the second one is always positive, meaning that the estimates here, yeah, there is a slight downward bias for the genotype variance, a slightly upward bias, and genotype by location variance, but it's not significant. So the estimate, the confidence limits include the 0, so the estimates are not significant different from 0. With 10% genotypes dropped on a yearly basis and 2 years of testing. If we did the same with 90% dropout, we see that there's a significant genotype by location variance. This relative bias is 30% or 25%, depending on which selection, dropout, whether you have selection or whether you have random dropout. But what you can also see is that the trend here in genotype variance and genotype by location variance is the same. It's a downward bias for the genotype variance and an upward bias for genotype by location variance. Now, genotype location is highly significant, and it's a huge bias in variance components. It's 25%. It's a huge bias. What is the reason for this one? We also look at the variance components itself. We have run 1,000 simulations, and therefore we can look at each variance component, how often the variance component is estimated to 0. Have in mind that we use REML as estimation method. REML, by default, requires that the variance component is positive, which is reasonable because the variance is some quadratic form. But the ANOVA estimate could be negative. So REML estimates are, by default, can be biased or approximately unbiased only for large sample sizes. And when the ANOVA estimate is estimated negative, then the REML estimated, because it is required to be positive, this estimate is set to 0. So what we take a look at is the probability that the variance component is 0. Either it is estimated at 0, or the ANOVA estimate would be negative and it is set to 0. And we look at this probability for the genetic and genotypic and the remaining variances. And what you can see here is that the genotype variance is estimated or bounded at 0 in around about 6% of the cases. The genotype by location variance, where you see the largest bias, is in 25% in 1/4 of our simulations. And if you just ask the question, in how many simulation runs at least one of the variance component is estimated to 0, then you see that it's 70% of the simulation runs. So in more than half of the simulation runs, one of the variance components estimated in our analysis was bounded at 0. And this probably caused the bias, because what you can see here is that the bias in the variance components for the genotype, for the selection, and for random dropout, they are more like they are close to each other. And this bias was caused by the estimation method itself. So by using maximum likelihood estimation, so in our case REML, there's a high chance to, in our simulation, to have variance component bounded to 0. And this probably causes the bias if we have 90% dropout. If we look what happens if we have more years, so once again we look at 90% dropout, and what you can see here is a relative bias in percent. What we looked before is the 25 and 30% of the bias for the genotype times location variance. And what you can see on the y-axis is the number of testing years. And the quarters are the genotype or location variance. The genetic variance are the polygonals or these signs are the genotype by year variance component. And the asterisks indicated here, you see two asterisks. This indicated that the variance component was significantly different from 0. And what you can see is that the relative bias decreases with more testing years, and that the significant results reduced, and in the last year they were very close to 0, and none of the variance components was significant from 0. So even with 90% dropout, where we observed the largest bias, we see that the bias reduced with more testing years, and more testing years means that I have more information to estimate the variance component. The variance component estimate itself gets more precise, and if the estimates get more precise, the chances that the bias, that the variance component, the ANOVA estimate, gets negative, and therefore the REML estimate was bounded at 0, decreases. So the reduced bias here was caused by the reduced chance that the variance component was biased, bounded at 0. For this, I want to make in the end, a conclusion. So is there just want to ask my original questions. Is there a bias in variance component estimates? In contrast to our paper in 2006, I have to say, yes, there is. We did not see this at that time because our variance components were not so small, the genetic variance components were not so small, and the dropout were not so large, so that we didn't see this. The second question, as the other paper said, it depends on the selection. Is it caused by selection? And there I have to say, no, it's just a property of the estimation methods we are using, the REML algorithm we are using. And this algorithm is consistent, meaning that it's asymptotically unbiased, but in small samples, small data sets, there could be a bias, and that's what we observed in our simulation. With this, I want to give the references. It's a 2006 paper from our group, and the Aguate in 2019, and the work will be published in also Crop Science in 2021, hopefully, because it's already in revision. Thank you for your attention, and if there are questions, feel free to ask them, and I'm happy to have a good discussion.
Yeah, thank you very much, Jens, for the interesting talk. So if there are any questions, either drop a message in the chat or you can also unmute yourself. So there's one question from Letizia in the chat. I can just read it out loud if you don't mind, Jens. So Letizia asks, in your table 3, why the bias is higher for random scenarios than for blood-based scenarios? I would expect that selection could induce more bias in the estimates.
Yeah, that's a good question. The two scenarios, if we have what we did here is blob-based selection. So the genotype selected by blob. This does not matter whether it's blob or blue. But if you have 90% dropout, then the best 4 genotypes are selected in the first year. And there's a high chance that these genotypes are also selected in the second year, or maybe 2 or 3 out of them. And if a genotype is very good in the first year, it will be selected in the second year, and the third year, and the fourth year, and the fifth year. So there's a high chance that this genotype, even with 90% dropout, remains in the data. If we use random dropout, the 4 selected genotypes are probably not selected in the second year, meaning that bridges from year to year only depend on the 4 genotypes staying the same in 2 years, but there's no connection between the first year and the third year, probably. So the structure of missing data is different in the genotype, in the blood-based selection, or in the random dropout. So if we have luckily the best genotype in the first year, there's a chance that this genotype allows us to compare and adjust the different years directly through this genotype, while when we have random selection, it's really rare that this genotype remain in all years. So if we compare first year to third year, we have to connect the 2 years by using the second year. And this slightly increase or makes the variance components a little bit more unstable. And that's why, sorry, you look at this one. And this makes the variance components slightly more unstable. And variance components more unstable means that there's a higher chance that the variance component, the ANOVA estimate of the variance component is negative, and the REML estimate is bounded at 0. That's why you can see that the under random selection, which are the black signs, are always more distance from 0 than the enlightened, the white signs here. And this is, I think, what the question was about.
So sorry if I just jump in with, from what I understand, basically, so you're saying, or you're concluding basically, you should not ignore missing at random data, but it's depending on in which year you are from your trial. So basically, the longer your trial, the more you can ignore it, basically. Because as you said in your first paper from 2006, you just said in general, you can just ignore missing at random data. But now it's, as you said, the picture isn't very clear. So if you're at the start of your trial, it makes or you have an effect of this missing at random data, and the longer the trial goes on. Is this correct?
Okay, maybe I didn't make this point clear enough.
No.
I think you can ignore the selection process completely.
Okay.
Not selecting, making a random selection is not a valid option in plant breeding. So this is just for simulation purpose that what we did. So And from my point of view, the bias in random selection and in blood-based selection is more or less the same. It's slightly higher under random selection because the connectivity between the years is reduced because genotypes are randomly dropped in each year, while with blood-based selection, the best genotype could remain across 8 years. So the connectivity between years is slightly better, which increases slightly the precision of variance components. But in both cases, you can simply ignore the missing data process because the variance, the difference between these two as the missing data pattern is very small compared to the bias you observed here. So the bias mainly depends on just using maximum likelihood estimation methods. And if you are in a normal range of variance components, and in a normal range of a yearly dropout of genotypes, I guess in most cultivar evaluation trials, you just drop out 50% of the data. Then there's no problem in estimating variance components.
Makes absolutely sense. So there are more questions from Gregor. So Gregor asks, have you looked into the accuracy of estimated genotype values using true or wrong variance components? It's the genotype values that matter in the end. Do you want me to repeat, or can you see if you look into the chat, the second question?
Yeah, I just, the second?
Yeah, Gregor's question.
I'm not sure whether I got the question because here you can see the precision of the variance component estimates. And what we did is that we increased the genotype variance. So maybe I have to tell a little bit more about the data. It's a data set from Argentina. It's a randomized complete block design in each location. But what they do in the analysis, they just ignore the block effect. Ignoring the block effect will increase the error variance. Furthermore, the genotypes are grouped in 8 homogeneous groups, and in which in each group they estimate genetic variance. So the genetic variance is properly decreased compared to the whole set of genotypes. And this increases the ratio of genotype to error variance components, making the genetic variance very small and the error variance much larger. And therefore, what we did is we increased the genotype variance by a factor of 10. In our simulation 2006, it was around about 5 times larger. And if you increase the genotype variance, the probability that this genotype variance is bounded at 0 is nearly 0, and the bias is gone. So increasing the genetic variances will decrease the probability that the variance component is bounded, and then you don't have any problem in the estimation method and no bias in the variance component estimate. I don't know whether this answers the question.
If Gregor wants to follow up, you're welcome to unmute yourself or just drop another message in the chat, Gregor.
Yeah, it's just, you are talking about the estimates of the variance components. That's fine. I'm just wondering if I use biased estimate of the variance components, how much would that impact the estimate of the genotypic values? Effectively, am I going to be doing wrong selections based on those estimates?
I've not looked on the final estimates of the genotypes itself. I've not checked whether the selection itself was correct. I just was looking on the variance components. I'm not sure how large the effect on the genotype effects will be, because, for example, for blobs, there's a regression to the mean, but the the testing intensity is the same for all genotypes in my scenario. So probably the ranking of genotypes, the estimates will change because the regression of the blobs will change, but the ranking will not change due to wrong estimates on the variance components. But estimates on variance components are used, for example, to calculate heritabilities, to calculate reliabilities, and for these to evaluate the quality of a testing system. For these things, variance components are important, and there, if you have a bias in variance component, you may do different conclusions on your precision.
Thank you, Jens.
There's another question from Salam. So first of all, he writes, thanks you for your nice presentation. We would say that bias is basically the reflection of data size instead of the REML method itself. I mean, lack of data cannot be compensated even with more sophisticated methods. What do you think?
It's there's a slight Bayesian point of view in this question, I think. Obviously, we can simply use an ANOVA method, and then we have an unbiased estimation on the one side. But REML itself is also efficient, meaning efficient that the estimate has smallest possible variance. So there are good properties of the REML algorithm, and there are good reasons that the REML algorithm is used, especially in big, large data sets where you have selection. Note that you can select genotypes from step 1 to step 2 and still ignore the process, the selection process, as missing at random can be ignored. Yes, there's a drawback of REML that the algorithm method itself is for small data biased. Yeah, it reflects in some sense the uncertainty or the incomplete or the small data sets. In some sense, this is right.
Thanks. There are more. Sorry, I just tried to stay on top. So there's a question from Daniel. He asks, have you looked at Robin Thompson's 1973 paper? Might be illuminating in terms of estimating variance parameters in the presence of selection. I don't know whether you know the paper.
I have no idea about the paper.
So
thank you for the hint. I will take a look on this.
There are more questions. So Renzo asks or says, thanks for the interesting talk. Maybe I missed this or it was already mentioned. If I got the second last slide correctly, you mentioned that in the paper of 2006, variance components were not small, thus bias was not observed much. Would this imply that REML has higher chance to give bias estimates if variance of the trait are close to 0? If so, what can be the impact on highly selected lines where variances become lower due to selection? It's a long question in case you want to read it yourself.
Yeah, no, I just read while you're talking. It's an interesting question. First of all, you're right that if variance components are in a usual range of evaluation trials, there's a paper from Leidig 2008 where he analyzed German evaluation trials across 30 years and give variance components for different cultivars or different crops. And whenever you are in the range of these variance components, this is what I mean that in a normal range. And if you are in the normal range, from my point of view, and if you only select 50 or 60% of the data, not 90% of the data, then everything, the REML estimate will be fine, and it would be hard for you to find that there is some bias in the variance component estimation. And that was the reason why in 2006 we see nothing in our simulation. The genetic variance will decrease during your breeding process, probably. And you're right, if the chance of the variance component to get bounded increase, the bias will increase. But this does not mean that the genetic variance itself decreases. It's more or less the ratio of the genetic variance and the error variances. So in some sense, the variance decreases, but from my experience, the variance in the breeding process does not decrease that much that it is relevant, maybe factor 10 or more. I expect that the variance, the genetic variance, will not decrease that much.
Okay, then there's a question from Felipe.
Thank you for the question.
So he says, I ended up entering the presentation a little late, so I'm sorry if this was mentioned and I missed it. My question, have you compared variance components estimated via REML and MCMC algorithms? I did not perform any formal simulations for testing that, but at least in the very balanced data that I have the opportunity to analyze, algorithms based on MCMC show better results in terms of convergences, bias, non-zero values, and other metrics.
I've not looked at Markov chain as a MCMC, so I've not looked at this. So cannot compare the REML algorithm with these properties. So
okay.
No idea. Sorry.
And then there's another comment from Daniel. So he says, I am not sure I agree with your statement that REML estimates of variance parameters must be positive. The issue of positive estimates arises through the choice of method to solve the score equations, typically taken as a qualitative, as you mentioned, but this should not translate to the choice of REML means positive estimates. A subtle yet important difference, I would say. Yeah.
I'm not sure whether I was imprecise in my
you mentioned in
my
your talk.
presentation.
Sorry, I'll just speak in. Sorry,
thank you.
Thank you, Daniel.
Firstly, very nice presentation. I was looking forward to this one, hence why I sort of asked Hans Peter to get someone from his group to present. So very good and very good stuff, right up my sort of alley in biometrics. I think the first thing that I mentioned was about Robin's paper in 1973. And so that one would be really relevant to the sort of the work you've been doing here as well, even if from sort of more of a historical perspective. This whole issue of estimating variance parameters when you've got records subject to culling is the way that sort of he put it. So that was, again, just more of a recommendation. And the second comment was, again, just me being a bit nitpicky, I think, in that I said that your statement, you sort of said that by default REML estimates of variance parameters must be positive, and sort of went on about, you know, ANOVA choices can go negative, blah, blah, blah. I was just sort of saying that, again, it's not really about REML forcing them to be positive. It's the way that you solve your score equations and get your estimates. That's usually taken as a Taylor series that uses a quadratic. And again, just a bit of a subtlety, but just something worth mentioning as well.
Yes, you're right. You're right. The result of a REML estimation method is a positive estimate, or zero if it's bounded to zero. It's not. There's no chance that the REML estimate will be negative. That's what I want to say. Does it fit to your definition?
Not really, because I'm sort of saying it's because the way that you estimate it with REML, it's not so much about the REML estimate. Let me just think about a good way to put this. it's not so much about the REML, you know, the algorithm itself, but it's the choice in which you solve the score equations. And so typically it's taken as a quadratic, like you were sort of saying, but there's no reason that you can't actually use a cubic or a higher order in the Taylor series.
Okay, yes,
And
you're right.
Hence, from that, you can actually obtain negative estimates, basically, is what I'm getting at. And so there was a subtlety there that, yes, by default, it's taken as a quadratic. It will be either 0 or positive.
You're right.
But if you choose to use a higher order in the Taylor series, you end up getting a different form of your, you know, your score equations in the way you estimate it. Sorry, the way you solve that, you end up getting negative estimates. And it was just worth mentioning because I'm not sure, I haven't read the preprint or anything like that, but it would be a criticism to say, you know, that you can't get negative estimates when actually you can get negative estimates from REML.
Yes, you're right.
Again, just a comment. Sorry, being nitpicky, but if you're
no, it's absolutely fine, and you're correct. I just look at the default method, which is also implemented in software, and there it's just a quadratic form. If you just run procmix, for example, or REML, then it's using REML and it's using the quadratic form.
Exactly.
And there it's positive.
Completely agree with you on that. It's just that in some of the software, ASREML is actually one of them, is that you can obtain negative estimates. So you still have the functionality to obtain the negative estimates in ASREML. You can, yes. And so the way about doing that is you just switch the bound from that being a P to a U, and you can obtain it. Although, as you sort of mentioned, small data sets and stuff, you probably wouldn't do it because you can have convergence and all the rest of the issues, blah, 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 but you can actually do it. In ASREML, you definitely can.
Maybe you can do it the same in SAS because there's a no bound option too.
Possibly. I'm not sure about that.
But I didn't know what the procedures are doing then.
Yeah. So,
but the default is just that it's positive, and you normally don't see that the variance component is negative, and so again, therefore you just see the 0.
Yes, again, completely agree with you, but there's a little bit of a functionality. It's one of those things that if you don't know about it, you may never, because ASREML was a bit of a, well, we all know what ASREML was like. So, but nonetheless, very nice talk, and thank you for joining us down here at Rosalind to give that talk too.
Thank you for your comment.
No worries.
Thanks, Daniel. So as far as I can see, there are no further questions or comments. And so I would end the meeting for today, and then the next seminar is in 2 weeks, and we are going to send an invite again next week with announcing the speaker and the topic. And good to see everyone today, and thanks for joining, and thank you again, Jens, for agreeing on giving this interesting talk.
Thank you for the invitation.